everyone. Let's start with the cell signaling course, the next class, where we are going to understand how receptor-like kinases signal to shape plants. What we noticed in the previous class was that how receptor-like kinases work, what pathway they activate, what are the signals over there, and how different signals engage with receptor to mediate through MAP kinase pathway different aspects of the development. In this regard, we will take some example today where we'll see how receptor-like kinase signaling is shaping the different plant growth aspect. One other thing which we would touch upon is root development or root hair development, shoot development, particularly with respect to the apical meristem, whether it is root apical meristem or shoot apical meristem. And then we will see that how a spatiotemporal signaling is required to shape the stem cell pool as well as ensure that the mature cells are there and they are acquiring a fate of differentiated cell. Similarly, we will see that how different RLKs, receptor-like kinases, the secreted peptides, which are the ligands, and receptor-like proteins, they participate in this process. We will also touch upon the stomata development aspect, which is mediated by the coordinated RLK signaling. So let's start with the shoot apical meristem. What exactly this area is? This is the apical meristem, which you will see in the plant apex. If you go inside, you would realize that this particular region can be further identified as something peripheral zone. Then there will be a central zone. And then just beneath the central zone, you have something which is called organizing center. And if you look at the arrangement of cells over there, they are arranged in multiple layers, and we are looking at these three different layers, L1, L2 and L3. We will see how different receptors engage with the secreted peptide to ensure that stem cell pool is maintained, and the organizing center which is going to instruct that what becomes the peripheral zone and what becomes the central zone. This is very important because something which remains in this central zone maintains the meristem identity, whereas something which is pushed into the peripheral zone will acquire the leaf identity. Also, at some point of time, some of these peripheral zone may engage into a very distinct activity through these receptors and make it a bud or flower. So what is seen here is that multiple different types of RLKs present in peripheral zone or present in the central zone. They identify the signals. So this is a signal which is here, and this is another signal. These are under the class of these molecules which are called clavata, and they have been identified by virtue of certain mutant analysis where this region is having a club shaped appearance, no leaf coming out, no meristem being maintained, and so on. So this is referring to the clavata. You will see that multiple different clavata-like genes are here. CLB3, which is a secreted peptide, CLB1, which is one of the receptor, and CLB2, which looks like a receptor-like protein. If you recollect from the previous class, together with RLPs and RLKs and RLCKs, receptor-like cytoplasmic kinase, the entire signaling downstream of the receptor is activated. And if you also recollect, we saw that plants largely respond by receptor-like kinases, where almost 600 genes are there which code for the receptor, and almost 1000 genes are there which are involved in this peptide-like signal formation. So normally the peripheral zone is responding to this peptide, CLE peptide, and sends the signal to the stem cell pool to maintain the stem cell nature. And if you look at the central zone, the clavata 3-like peptide, it engages with its receptor in combination as a heterodimeric receptor between clavata1 and the BAM1 and 2. You will probably understand in many different contexts that how these genes are named in any plants or any organism where these mutant phenotypes are analyzed. Based on what exactly you get as a mutant phenotype, genes are named. Like here, BAM is called barely any meristem. So the gene which is leading to this phenotype is named as BAM. So together with the different receptors which are present in this region, you will find that the CLV3 peptide binds, and together they instruct this pool, the stem cell pool, to be localized in this area and remain inhibited. The regions which are the cells which are moving in the peripheral zone or moving upward, they acquire the meristematic property and then divide, and the stem cell pool is maintained here. And this is basically because of different signals, different receptors, and something which is like receptor-like protein, or something which is like a receptor-like cytoplasmic kinase. What you notice here in all these cases, there is a functional engagement of different components of this receptor-like kinase pathway, which ensures that the intracellular region is activated by phosphorylation, which exerts its function. So this apical meristem region which is being organized is by virtue of different molecules which are part of the receptor-like kinase signaling. We saw these molecules in the previous slide. These are different zones where expression are happening. Further analysis has suggested that there are distinct domains within this apical meristem region which is important for maintenance of shoot apical meristem. Apart from these different molecules, one very important gene, which is WUSCHEL, is engaging in interaction with CLV3 and these CLV2 like molecule, and ensuring that the signaling by RLK is activated. If you recollect, the signaling is activated by engaging into this MAP kinase pathway where 3 different kinases are there, and they initiate a cascade of phosphorylation event where for MAP3K, where MAP3K activates MAP2K and then to MAPK. Now if you look at the root apical meristem, where again a set of these molecules which are in the class of receptor-like kinases, they are involved in differentiation and development of root apical meristem. In this meristem region, you have distinct zone where different molecules or different components of receptor-like kinase signaling are activated or present. They have a expression domain and where they ensure activity is being maintained. So here you have this secreted peptide CLE40, which signals through this ACR, which is a receptor-like kinase, which helps in the maintenance of stem cell niche. Here we are discussing in particular the cells in this region which are called columella cells. So what you notice in this root apical meristem, that there are distinct zones which are identified by the expression of very distinct molecules which are part of receptor-like kinase signaling. This is secreted peptide, this is your receptor-like kinase, and then you have these receptors as well as the ligand, which ensures different regions in this root apical meristem. So what you would notice here is that the cells which are there in the root apical meristem region, and if you talk about this particular domain which is WOX5 expression domain, this expression ensures that this region remains as a quiescent center. If I say QC, quiescent center, basically the cells here, they are not actively dividing, but the regions around it, they are actively dividing. And the signal from WOX5, which is promoting not only which is promoting the quiescence activity in this region, but also promotes the stem cell niche in this region, which is ACR positive. So together with the action of ACR4 and WOX5, the columella cells in the root apical meristem will maintain a apical meristem or stem cell identity, or they will undergo differentiation and ensure that the other identity of the neighboring cells that can be acquired. So what you notice is that again receptor-like kinase signaling is playing very important role in the root apical meristem development as such. Now if you think about another aspect which is associated with root, and that is root hair development. If I look at the previous, go back to the previous slide, this region which you see in the proximity here, not necessarily in the root apical meristem, but on the lateral sides, these are basically the epidermal cells. And from these epidermal cells only you will have a particular cell which is differentiating as a root hair cell. So if you look at the cross section of a root, this is the outermost epidermal cell layer, and below that you have cortical cell layer or cortex cells. If I can just make sections in this region indicating different cells which are part of epidermal area, and I mark the regions which are inside the cortex region and see what kind of cells are there. So what you can identify that there will be two distinct types of cell population here. Like if I can mark one of those cell types, so if you think that this epidermal cell, remember that this epidermal cell only will become the root hair. It is in direct contact with the cortex cell. So see this scenario, consider this as your epidermal cell layer. There are two cells which are part of epidermis. Now there is a cortex cell which is above it. So this is cortex cell and this is epidermal cell. What has been identified that there are cells which are under directly a single cortex cell, and there are the epidermal cells which are under two different cortex cells. This positional signal itself is good enough to make them undergo certain transcriptional programming which will ensure that the cell which remains under a single cortical cell acquires a non-root hair cell identity, and the other one which has seen contacts with two distinct cortex cell makes this particular thing active, which is root hair development, and acquires the root hair cell identity. So if you look more carefully inside what happens over there, here again there will be signals which are coming from these cortical cells, and since we are discussing the receptor-like kinases, you will realize that these receptor-like kinases will require their compounds to which they are responding. So there will be these peptides, and also if you recollect, there are plant hormones which are responding. So you have brasinosteroid also signaling, and this particular case of root hair development gives us classical example of inhibiting a particular program which allows the next program to be activated. So as we saw in the previous case, the cell which is under one cortical cell is non-root hair cell, and the one which sees two distinct cortical cell acquires the root hair cell identity. So the receptor-like kinases of this family, as well as something which is responding to the brasinosteroid, they give signal to both these cells. So there is a transcriptional network which sets in because of these receptors responding to these ligand which are being released by cortex. People still are working to identify what is the exact nature of these signal, but what is understood is that these signal bind to the receptor, they activate a program inside this cell which inhibits root hair cell fate. So positional identity tells them that activate a program which will inhibit the root hair cell. Now the other cell which is here, which has the similar program set in, but the neighboring cell sends a inhibitory signal not to activate a transcriptional network which inhibits the root hair cell formation. This is where in the previous slide we saw root hair development, a transcription factor which will induce the root hair path. So what you realize that adjacent cell, this responds so that there is no root hair cell fate. It also sends a signal to the neighboring cell which is under two different cortical cell to not activate the transcriptional program which stops it from being a root hair cell. So basically inhibiting an inhibitory signal, thus activating the signal, and what is being activated here is the root hair cell fate. So you realize that how different kind of signal, different types of receptor, like this receptor called BRI or brasinosteroid insensitive, and these two receptors, they identify different ligand and set in a program which together works to ensure non-root hair cell fate and root hair cell fate. Let's take another example of stomata development. We all understand how important is stomata for various processes of plant growth. This receives signal in different forms, brasinosteroid. There are epidermal growth factor kind of molecule signals which ensure a signaling, and then there are signals which are called stomagen. So basically stomagen will like to activate more stomata formation, and these stomata cell, they actually come from the epidermal cell modification. So what is understood that under normal circumstances these epidermal cell will remain epidermal by virtue of the signaling by this epidermal promoting factor 1, which works through this receptor-like kinase. This receptor-like kinase responds to this factor, activates a pathway which internally involves many more molecules. So in this step you can have RLK kind of molecule which eventually will phosphorylate this molecule, and YDA is actually your MAP3K, which is the first molecule to be activated in MAP3 kinase pathway. So once MAP3K is activated, it activates MAP2K, which in this case is identified as MAP kinase 4-5. In previous class I told you that these MAP3K, MAP2K, MAPK are class, or in previous class I told you that MAP3K, MAP2K, MAPK are a class of proteins. One of those proteins, for example, here is called MAP kinase 4-5. This is in the class of MAP2K. So once this is activated, this will take the next target, which is MAP kinase MAP36. This is the last molecule in this signaling pathway, which is MAPK type. MAP3K, MAP2K and MAPK. Now this molecule through intermediates stops this transcription factor. What this transcription factor was mediating was conversion of epidermal cells into a cell which will make the stomata. So normally in absence of any of this stomagen kind of signal, epidermal fate is preferred and this epidermal cell is not converting into stomata. But if there are overwhelming stomagen, it will inhibit this signaling. What it will do, inhibition of this signaling, meaning that this inhibition on the transcription factor is lost, because activation of MAP36 was inhibiting this transcription factor, which should have promoted the stomata formation. So now if you inhibit this MAPK36, you will see that this inhibition is no more there, and that is why you will see more and more stomata formation. So you'll realize that relative abundance of these signals drive the signaling in one particular direction and allows fate change of a cell from epidermal to meristemoid or stomata type cell formation. There is additional regulation. Remember that all these plant tissues are receiving multiple different types of signal, and eventually it has to come to a conclusion where some of these molecules are widely regulated. This YDA, the MAP3K, is very commonly regulated by multiple different signals. One of them is this, that where brasinosteride signaling activates a phosphatase, which will activate YDA. What basically this will do, once YDA is activated, this signaling pathway is activated and there will be no conversion of epidermal tissue or epidermal cell into a stomata cell. So realize that, so you would have realized that how different kind of signaling is working together to ensure that the signaling is moving forward in the desired direction. What you can understand further is that this is not only one example which I gave you for YDA, there could be multiple upstream growth and developmental cues. Think about a process, you will find that there are definite set of signals, secreted peptides, brassinosterides, they are coming together and they are ensuring that final aspect of that signaling is met. What you are seeing in this slide is classical example of how this particular MAP kinase pathway, where YDA is MAP3K, this MAPKK45 is 2K and MAPK36 is 1K. So whatever signal is coming from different routes which involves next set of molecules at the end to mediate a particular process like shown here. So before the signal terminates into YDA, it must have originated at the plasma membrane where a receptor found its ligand and it started the serine threonine kinase activity by different routes which we discussed earlier, different modes of signal engaging with the receptor. And then you will probably see here that you activate a RLK downstream of the receptor, which is key molecule in phosphorylating the MAP3K. So receptor binding to its ligand, activating an RLK, which also needs to be phosphorylated, and this activated RLK activates the MAP3K, which follows the cascade of phosphorylation event. So if you take example, we saw in the previous case that in the stomata development you had this molecule called speechless. Then in case of pollen development you have this class of molecule which are called Werke different types, because you see Werke 34 is working in pollen development and Werke 2 is working in the embryonic patterning. Similarly, you can have multiple other different types of final regulator or effector, like here AGL15, which is important for the floral abscission. So in some sense what this slide is showing you, that multi-to-one approach. If you remember, we saw that multiple kinases, if I say K1, K2, K3, K4, K5, through the RLCKs, they are merging onto this final molecule, which is your YDA. So many to one signaling is happening here. We have seen multiple examples of this sort where multiple different kinases are working to ensure that one key transcription factor or one key effector molecule is activated. In this case, which we saw earlier, whether it was stomata development or embryo patterning or pollen development or floral abscission, all of them were from their receptor, they were merging onto this YDA molecule, which needs to be phosphorylated. This takes to the next molecule, which was MAP2K5645. This will be phosphorylated, then it will take it to the MAPK36, which will be phosphorylated. And this phosphorylated final molecule, if you recollect in the previous class, I told that not necessarily this final MAPK is your actual molecule. Sometimes this may enter into the nucleus and perform the activity directly over there, or this will activate a next molecule. We saw in one case speechless, which is a transcription factor, and this is important for the stomata development. So what we have understood in this class, that whether it is root development, shoot development, stomata, or the root hair development, all are tightly regulated by signaling through this receptor-like kinases. You will need a complex mix of secreted peptide. You saw CLV40 peptide in one case, CLV3 peptide in one case, CLE peptide, as well as steroid hormone, which we have been seeing in family of brasinosteroid. These combinations work together through different receptors to ensure that final response is mounted. What we also notice that inhibitory feedback while integrating different signals is very important to ensuring that proper control is exerted. What I mean over there is that the speechless, if you recollect, was inhibited by MAP kinase signaling. And this will not allow stomata formation. I'll say this inhibition, you have no stomata. So when you need stomata formation, you need to inhibit this signaling. And this is what stomagen was doing. So all in all, receptor-like kinase signaling shapes multiple different aspects of plant growth, whether it is root apical meristem, shoot apical meristem, stomata, or root hair development, or we saw multiple other cases earlier that pollen development and so on. Embryo patterning is there. So receptor-like kinases are the workhorse in plant when it comes to signaling, and plants are heavily dependent on receptor-like kinases are very important to be important for plant signaling events which are happening. Thank you.